Hello, in this video we will discuss about the physiology of peroxisome proliferated activity receptor alpha. So what is this? And basically we will learn about the whole story of the PPAR alpha. So first of all we should need to understand during the fat state when we will eat something. For example, carbohydrate, fats, lipid, protein and amino acid. So when we will intake is known as ingestion. After the mastication go into the stomach is known as bolus, as a food, and the digestion will begin after the digestion, partial digestion of carbohydrate in the oral cavity. It will move into the digestive system for further digestion of the protein, and the semi digestion will occur in the stomach, will go into the duodenum, the small intestine, to become a chyme. And the chyme is basically the digestion will occur more, and in this way the complete digestion will occur in the duodenum, in the small intestine. So also absorption will occur. So this is the ingestion to digestion, after the digestion into the absorption into the blood circulatory system. So anyhow, this is the intestine and this is the chyme, and basically chyme contain dietary fat if and dietary glucose. Let's suppose, so it will move into the blood circulatory system. When go into the blood circulatory system, the hypercholesteremia, the hyperlipidemia and the hyperglycemia will occur. So remember, after this, the increase of the glucose level, increase of the lipid level will lead to low density lipoprotein increase, but it will be the higher glucose level will trigger the beta cell of pancreas to release the insulin. While in this way here is the adipose tissue will be not lipolysis but the lipogenesis will occur due to the insulin production. So this is the beta cell which that require the GLUT2 channel will allow to influx of the glucose due to the hyperglycemia, will stimulate the beta cell to exocytose of the vesicle containing the insulin into the blood circulatory system, and it is the endocrine hormone. So this is the pancreas. After going to the blood circulatory system, this is the liver. So the liver contain insulin receptor will bind with the insulin, will lead to open the GLUT4 channel for influx of the glucose, and it in this way this is the facilitate transport. So after this, glucose will move inside, will lead to go the glycolysis to produce acetyl-CoA, and the acetyl-CoA used in the mitochondria, and on the other hand the lipogenesis will be occur. The lipogenesis is a long process. Here is the insulin, which that is the lipogenesis will be occur and triacylglycerol formation, but the glycolysis is the long and the lipogenesis is the long steps. I am missing here because we should don't understand here, but separate video I will understand you. So this is the free fatty acid will be formed, and in this way the free fatty acid is also through glucose but also from the directly from the fatty acid into the liver go through a portal vein. So here is the DGAT and ATGL is required for reversible to produce triacylglycerol, triglyceride storage in the liver also triggering. And in this way here is the PPAR alpha are present in the nuclear receptor, and here is the nucleus and contain DNA. So remember, this will be stimulated through fatty acid, and why the stimulation will required and how the stimulation will change the physiology we will discuss now. So the FGF21 will be increased due to the transcription to translation from the nucleus through a PPAR alpha. It means that directly trigger the FGF21, and also it enhance the beta oxidation, because FGF21 is required for the beta oxidation, and the transcription to that protein enzyme will synthesis and produce through translation which that required for beta oxidation and acetyl-CoA formation, will lead to ketogenesis also, ketosis, and in this the gluconeogenesis will also occur without fasted state. Even the fasted state will require these things, but the ketones body and the glucose require the energy in the peripheral tissue. So remember this character is also the part of the fasted state, but during the fat state the PPAR alpha is triggering the beta oxidation of fatty acid because it is important for a normal physiology. So let's begin to understand the fasted state. So the fasted state, the sympathetic nervous system increase during the not rest and digest, but the fight and flight response will lead to adrenergic nerve stimulation as the nervous system, while the endocrine hormone will stimulate through pituitary gland and through hypothalamus to the pituitary gland to produce the corticotropin releasing hormone, which that will be corticotropin hormone, which that will target to the adrenal gland to enhance the glucocorticoid and adrenaline increase. So the glucocorticoid and adrenaline when increase go into the blood circulatory system and will target to the adipose tissue, and target to the adipose tissue when bound with the epinephrine in the epinephrine receptor or glucagon receptor. In this way the epinephrine is also go into the alpha cell of the pancreas, and it will be trigger the triacylglycerol to convert the lipolysis will be occur and free fatty acid and glycerol formation through a hormone sensitive lipase enzyme. So anyhow, here is the blood circulatory system. The epinephrine will also bind as a sympathetic nervous system response, and the catabolism will begin due to the glucagon production, and the glucagon will increase in the blood circulatory system while the insulin will inhibit. So in this, the glucagon is very important for enhance the gluconeogenesis and the glycogenolysis and other catabolism, the proteolysis. So in this way, here is you can see this the glucagon will trigger the protein kinase A activated enzyme which enhance due to the molecular pathway, and in this way it will be target to the PPAR alpha, the nuclear receptor, which that will trigger the same mechanism to beta oxidation and ketogenesis, the gluconeogenesis, and ketone bodies and glucose formation. While on the other hand, here is the free fatty acid will also the part here you can understand. Increase will lead to increase the PPAR alpha. And on the other hand, here is a mTOR C1 complex will decrease due to the fasting state, will trigger the PPAR alpha also, and the AMP adenosine monophosphate will increase due to the fasted state, will trigger the AMPK pathway increase, will lead to also trigger the PPAR alpha. So the PPAR alpha is used for the beta oxidation and catabolism and the gluconeogenesis and the ketones body production, and in this way the keto adaptation due to the PPAR alpha is very important. So here is the signal, for example, when nuclear receptor will receive to give that signal and transcription to translation to function. And what is the function? Let's begin to understand the whole process and the function and mechanism. So let's begin to understand that is now I am sketching the chart for understanding the better the function of the PPAR alpha, the peroxisome proliferated activated receptor. So the function is the lipid regulation also. Number one is the promotion of beta oxidation, serum triacylglycerol clearance, and high density lipoprotein increase. So that is beneficial. And induction of hepatic lipogenesis will also occur, the lipids formation in the liver. While on the other hand, the glucose regulation also occurs due to the PPAR alpha, the alleviation of glucose disposal in skeletal muscle from the blood circulatory system, and that is why the disposal of the glucose into the skeletal muscle for better use during the exercise and fasted state. And the number two is the amelioration of insulin resistance and decrease the aerobic glycolysis in cancer cells. So third one step. Amino acid regulation, the number one is the process, the amino acid regulation, the decrease of amino metabolism rate as a part of starvation response. So in this way, the amino acid will not break down of the protein, so the survival and the protein will be maintained, and the while the glucose will be used and other fat. So that is why the protein will not degrade during the fasted state. So the inflammation regulation is the number one is the gene expression of inflammatory cytokines. Example is the interleukin 6 and tumor necrotic factor alpha. And the number two is the down regulation of the APAF1 and the necrotic factor kappa B pathway for inhibition of the apoptosis and interaction with the glucocorticoid receptor alpha or estrogen receptor. And the cardiovascular benefit is that the activation of endothelial nitric oxide synthase will lead to vasodilation and lead to prevention of the coronary heart disease as well as other cardiovascular disease. So the tumorigenesis effect mean the inhibit of angiogenesis, the proliferation of the FAO2 glycolysis, the free fatty acid oxidation to glycolysis, and the disruption of the balance of glucose and lipids metabolism to inhibit ATP production. And the number four is the accumulation of reactive oxygen species in mitochondria and damage control the epigenetic also. The methylation of DNA will control. So this is a video about the physiology of the PPAR alpha, that is the peroxisome proliferated activated receptor, which that will lead to also cause the different function for the normal homeostasis. So the peroxisome proliferated mean the proliferation, the activation of the peroxisome proliferation, and the receptor are present in the nucleus, nuclear membrane. So that is why it is known as the nuclear receptor PPAR.